Good day, everyone, and welcome to Biochemistry for Medical Laboratory Science. Now, today, we'll be learning about enzymes. Now, enzymes govern all chemical reactions in living organisms. They are specialized proteins in its structure with fascinating precision and selectivity, which their main um, function is to catalyze biochemical reactions, which can store and release energy, can also make pigments in our hair and eyes, digest the food we eat, synthesize cellular building materials, and protect us by repairing cellular damage and clotting our blood. Now, enzymes are very sensitive to their environment. They respond quickly to changes in the cell. Now, the deficiency or excess of particular enzymes can cause certain diseases or signal problems, such as heart attacks and other organ damage. Now, our knowledge of protein structure can help us appreciate and better understand how enzymes function in living cells. Now, at the end of the learning session, the student must be able to understand concisely the function of an enzyme, explain properly the different models of enzymatic reactions, and enumerate completely the different classification of enzymes. Added to this, the student must also know correctly the different uses of enzymes. Now, let's first talk about the overview of their enzymes. Now, each cell in the human body contains thousands of different enzymes because almost every reaction in a cell requires its own specific enzyme. Now, enzymes cause cellular reactions to occur millions times faster than corresponding uncatalyzed reactions. Now, as catalysts, their enzymes are not consumed during the reaction, but merely help the reaction occur more rapidly. Now, until the 1980s, it was thought that all enzymes power proteins. Now, in the updates of your enzymes, a few enzymes po are now known that are made of ribonucleic acids. And as that catalyze cellular reactions po involving nucleic acids. Now, in this topic po, we will consider only enzymes that are proteins. Okay? So your enzymes po undergo all the reactions of proteins, including denaturation. Now, slight alterations in pH, temperature, and other protein denaturants affect also your enzyme activity dramatically. Okay, so also of your enzymes are globular proteins. So some are simple proteins consisting entirely of amino acid chains, and others are conjugated proteins containing additional chemical components. So these terms po we will tackle po on the succeeding slides. Okay, let's now learn the different terms when it comes to the structure of an enzyme. So first of this would be your simple enzymes. Now simple enzymes, when we are referring to its structure, it only composed of proteins. Okay, so your simple enzymes are purely are proteins. Okay, so your conjugated enzymes po in contrast, so it has a non-protein part in addition to a protein part. Okay? And your conjugated enzyme is just like a conjugated proteins, wherein there is a part of the structure that are non-protein po. And the non-protein part of a conjugated enzyme is called your cofactor. Okay? So your cofactor is the non-protein part of the conjugated enzyme. And the protein part naman po of their conjugated enzyme is called your apoenzyme. Okay? Together these two, they produce a conjugated enzyme. So why do apoenzymes need cofactors? Okay? So without the presence of the cofactor, your apoenzyme cannot be a functioning conjugated enzyme. So why do apoenzymes need cofactors? Now, cofactors provide additional chemically reactive functional groups po besides those present in the amino acid side chains of their apoenzyme. Thus, combining with it will form a functional conjugated enzyme. Okay? A full functioning conjugated enzyme composed of apoenzyme naman po and your cofactor will be called your holoenzyme. So holoenzyme is a biochemically active conjugated enzyme produced from an apoenzyme and a cofactor. So it's a combination of an apoenzyme and a cofactor entity. So ano naman po yung tinatawag na coenzyme? Now coenzyme po is just like a cofactor po. No? So it's just a cofactor but it doesn't have a chemically reactive functional groups to provide in your apoenzyme. So para lang din po siyang cofactor pero po hindi po siya nagbibigay ng functional groups. Instead, your coenzymes po can be a one element alone or one atom alone that is combined with your apoenzyme to turn on the function of the apoenzyme. Okay? So ang tawag po doon is a coenzyme. So one example of a coenzyme are your metals, okay? Your sodium, potassium, magnesium, and other metals that can activate your full enzyme. Okay, the next point of our discussion is the nomenclature and classification of your enzymes. So before we understand the different functions of your enzymes, we need first to know how your enzymes are named properly, okay, scientifically. So how our enzymes are named? So our enzymes are named about the function of the enzyme. So ginagamit po natin in the naming process is the function of your enzyme, also the type of reaction catalyzed by the specific enzyme, and also the identity of the substrate which your enzymes act upon. Okay? So your substrate po is called the reactant in an enzyme catalyzed reaction. So kumbaga po your substrate is the starting product in an enzyme catalyzed reaction. So to further understand the nomenclature of your enzymes, let us learn the three important aspects in the naming process of your enzymes. Okay, these are the three important aspects in the naming process of your enzymes. So first, most of your enzymes ending in the suffix ase or ase. Example of these are your urease, sucrase, and lipase. In the exception of your digestive enzymes, wherein your digestive enzymes use the suffix in or in at the end of its name. Example of this is your trypsin, chymotrypsin, and pepsin. Okay? So next naman po na aspect in the naming process of your enzyme is a type of reaction catalyzed by an enzyme. So usually po it is used as a prefix in the name of your enzyme. For example, so let's see the enzyme called oxidase. Okay? So usually, yung type of reaction catalyzed by the enzyme is placed at the prefix of its name. In the case of your oxidase, since this enzyme primarily um, catalyzes the reaction of called oxidation reaction, so its name is called oxy for its reaction, oxidase or oxidation, and then ase po pertaining to it as an enzyme. Another example of this is your hydrolase, which catalyzes the reaction called hydrolysis reaction. And kaya po yung pangalan niya is hydro and then lase. Okay? Hydro means hydrolysis reaction. Ase meaning it is a type of enzyme. And last important aspect in the naming process of enzyme is the identity of the substrate. Now, the identity of the substrate usually po is placed at the first part of the name of the enzyme followed by the type of reaction catalyzed. So kapag may substrate, kailangan may type of reaction catalyzed din. Example of this is your glucose oxidase. Okay? Ibig sabihin, this enzyme acts upon the glucose molecule using the reaction oxidation reaction, and then it is a type of enzyme. So another example is your pyruvate carboxylase, succinate dehydrogenase. Okay, let's now proceed now to the different classification of your enzymes. So your enzymes po are grouped into six major classes on the basis of the types of reactions they catalyze. So let's start with the first one. That would be your oxidoreductase. The oxidoreductase enzymes catalyzes the reaction called oxidation and reduction reaction. Okay? So please take note that your oxidoreductase always requires a coenzyme that is oxidized or reduced as the substrate is reduced or oxidized. Now, typically, these coenzymes are called your electron carriers. So examples of electron carriers is your NAD plus or what we call your nicotinamide adenine dinucleotide and your FAD, which is also called as your flavine adenine dinucleotide. So please take note again ha that your oxidoreductase, kapag po sila yung enzymes that acts upon a reaction called oxidoreductase or oxidation reduction reaction, always and always they needs a coenzyme to function. Okay? So next naman po is your transferases, which are enzymes that catalyzes naman the transfer of the functional group from one molecule to another. So the two subtypes of your transferases is your transaminases and your kinases. And transaminases po, these are transferases po that catalyzes the transfer of amino group from one molecule to another. Your kinases naman po, in the other hand, catalyzes the transfer of phosphate group from ATP molecule to give out ADP and a phosphorylated product. So itong mga kinases na to, they are always involved in the process of removing a phosphate group to the ATP or other triphosphates and then to give po a phosphorylated product and ADP. So it does that to produce energy po in return. Okay. In this slide po presents the example of a redox reaction catalyzed by your oxidoreductase enzyme called your lactate dehydrogenase. So your substrate here is your lactate. Ito po yung i-reduce na substrate or i-oxidize na substrate rather. And then this one, your NAD, is their coenzyme called an electron carrier. Okay? So ito po yung tatanggap ng hydrogen atoms later po na tatanggalin po sa inyong lactate substrate. Okay? To produce now your pyruvate, which is the oxidized form of your lactate, to give out naman reduced coenzyme and then an additional byproduct of hydrogen atoms. This hydrogen atom po is recycled lang by the system to be used by other reactions. Okay, this one naman is an example of a transferase reaction. Okay, catalyzed naman by your enzyme called hexokinase. So ang substrate mo dito would be your glucose and then the molecule na pagkukunan po ng inyong phosphate group na tatanggalin ng hexokinase would be your ATP. To transfer that phosphate to your substrate, specifically on this part, and then to produce a glucose 6-phosphate product. Okay? So ang inyong byproduct dito is a reduced or is a um, downgraded adenosine triphosphate into adenosine diphosphate. The next naman po would be your hydrolases. Now for hydrolases, it catalyzes the hydrolysis reaction. Now hydrolysis reaction, please take note that this is essential to the process of your digestion. So digestion of different biomolecules like your sugars, proteins, lipids, and other biomolecules. That's why there are in these subtypes of your hydrolases, it includes carbohydrases, proteases, and lipases. So for hydrolases po, it affects the breaking of the different bonds within the structure of your sugars, proteins, and lipids and other biomolecules. Now for carbohydrates, your carbohydrates affects on breaking of glycosidic bonds in oligo and polysaccharides. For your proteases naman po, it affects the breaking of peptide linkages in your proteins. And for your lipases naman, it affects on the breaking of ester linkages in triacid glycerols. Now, for your liases naman po, these liases po are enzymes that catalyze the addition of a group to a double bond or the removal of a group to form a double bond. But please take note that liases po um, exempt the principle of the hydrolysis and oxidation in the addition and removal of groups to create double or single bonds in return. Okay? Now, in this slide po depicts us the example of a hydrolysis reaction using a carbohydrate. Okay, so as you can see here, water molecule po is used to donate a hydrogen atoms here at the glycosidic linkage to break down these two glucose molecules in your maltose. Okay, this reaction naman po is a representative of a lyase enzyme wherein po the main goal of your lyases po is to create double or single bonds po in the structure of your molecule by removing different molecules po in the structure of your molecules. Now, the last two classifications of your enzyme is your isomerases and ligases. Now, for your isomerases po, these are enzymes that catalyzes the isomerization of a substrate in a reaction, converting it po to a molecule isomeric with itself. Okay? So, when we say isomerization, again, it is the rearrangement of atoms. So, please remember that in your isomerases, it only involves one reactant and one product in the enzymatic reaction. Okay? And lastly po for your ligases naman, these are classification of enzymes that catalyzes po the bonding of two molecules po into one with the participation of ATP. So this is an example of an isomerase enzymatic reaction. So wherein you use phosphoglyceromutase to arrange the position of the phosphate here at your substrate up to the second carbon. That's why the product of 3-phosphoglycerate arranging the position of the phosphate creates 2-phosphoglycerate product in return. Here in this slide naman po is a sample of a ligase enzyme called your pyruvate carboxylase, which combines your carbon dioxide and pyruvate in the usage of an ATP molecule to create oxaloacetate in return. So meron kami mga byproducts like your ADP and a phosphate po to be recycled po to another enzymatic reaction. Okay. Here in this table shows us the different subclasses of each main classes of enzymes. As you can see here, these are the subclasses of oxidoreductase. Added to this is your dehydrogenase or dehydrogenases enzymes. For your hydrolases, added to this would be your nucleases and phosphatases. Okay. In this table, for lyases, these are the subclasses, your dehydratases, decarboxylases, deaminases, and hydratases. For your isomerases po, dalawa ang subclass po niyan. This will be your racemases and mutases. So, ang difference po niyan is that racemases converts the DNL isomers of your molecules, while your mutases naman, if you need to transfer functional groups into one position or to another position, you use mutases. For ligases naman po, this would be your synthetases and carboxylases. Okay, let's now proceed now to the different models of enzyme action. Okay, now the different models of enzyme action are governed by the fact that the enzyme and substrate can be combined to form a complex. Now, before we proceed on that, your enzyme po kasi has a very specific part called the enzyme active site. Okay? So, the enzyme active site is a small part of an enzyme. Usually po, dun po nangyayari po yung mga catalysis po na ginagawa ng isang enzyme. It is a three-dimensional entity po formed by groups that come from different parts of the protein chains. Okay? So the explanation po of how enzyme function as catalysts in biochemical systems are based po on the concepts of an enzyme active site and enzyme substrate complex formation. Okay? So the enzyme substrate complex formation po is an intermediate reaction po wherein it is formed by the binding of the substrate in the active site. Then after binding the enzyme on the active site, as you can see here, the enzyme product complex naman will be formed. Now, this enzyme product complex um, uh, uh, is not permanent po, no? Ibig sabihin, the product is loosely bound to the active site. And then this loosely bound product in the active site can then be released on the enzyme. And then the enzyme po is not consumed to the reaction, kaya po siya yung ginagamit ulit to start the process over. Okay? So additional din po, no, the enzyme active site is also called as the crevice-like part or location in the enzyme. So study shows that only a small portion of an enzyme molecule called the active site participates in the interaction with a substrate or substrates during a reaction. Yeah, let's now proceed now on the presentation of the two basic models of your enzyme action. Okay, so first is the simplest of this model is your lock and key model. Okay, so the lock and key model just tells us the specificity of an enzyme to its substrate because of its rigid geometrical conformation of its active site. So this conformation po, geometrical conformation, can only complement with a geometrically complementary substrate that it can accommodate. Okay? So, ibig sabihin lang po, so may specific lang po na substrate ang kaya po niyang i-accommodate dun sa active site niya because of the geometrical conformation produced by the different R groups of its amino acid on its active site. Okay. Next model naman would be the partner of the lock and key model which is called your induced fit model. Okay? So induced fit model naman po, it shows us that besides of the active site being being specific to its substrate, the enzyme's active site also is flexible po. Okay, it is not rigid and static. So, pag sinabing not rigid, ibig sabihin, it is not rigid, um, hindi po siya conform to one shape. So, it can accommodate a large size of an substrate, and then hindi rin po siya static. Ibig sabihin, um, hindi po siya permanently bounded to its substrate. Okay? So the active site has constant changement or changing of the shape. So it allows for changes in the shape or geometry of the active site, okay, to accommodate a substrate. So it results po on the enzyme's flexibility and adaptation to the incoming substrate. So there are different forces that draws the substrate into the active site po. So like your same forces that maintains your tertiary structure in the folding of your peptide chains. Examples of these interactions is your electrostatic interactions, hydrogen bonding, hydrophobic interactions, which all helps po to attract and bind the substrate molecules. For example, a protonated or positively charged po na amino acid or amino group in a substrate could be attracted and held at the active site by a negatively charged aspartate or glutamate residue. So alternatively po, your cofactors po, such as positively charged metal ions, can often help to bind the substrate molecules to the active site. Okay. So this is a schematic diagram po that represents po the amino acid R group interactions that bind po a substrate to an enzyme active site. So the R group interaction that maintain the three-dimensional structure of an enzyme are also shown. Okay? So meron din po dito mga non-interacting R groups that helps naman on the solubility of the enzyme po. Now, let's now proceed now to the specificity of an enzyme. So, although your enzyme is specific to its product, so there are certain types din po of specificity of an enzyme. So, an enzyme can be very specific to one substrate, specific to a group of substrate. It can also be specific to the type of chemical bond or what we call your interaction, and then specific po to a type of chemical reaction. So the degree of specificity is determined po by the active site pa rin po. Okay? So enzymes exhibit different levels of selectivity. Okay? So some active sites can accommodate only one particular compound. So let's see po the different types of enzyme specificity. So first of this would be your absolute specificity. Okay? So pag sinabing absolute, no, such specificity po means that an enzyme po will catalyze a particular reaction for only one substrate. So it is the most restrictive of all specificity. Okay? So for example, your catalase po is an enzyme with absolute specificity. It can only act po on the separation of oxygen po, no, in hydrogen peroxide. Okay? So another is your urease, a bacterial inhibition, which is an enzyme with absolute specificity. So sabihin one reaction lang po and one product ang magkakaroon. Okay? Next naman po is a group specificity. So for a group of specificity, it acts only on molecules po that have a specific functional group. For example, a group of molecule has hydroxyl functional group. So all of that groups po, or all of that molecules is act upon by that enzyme. Okay? So one example of this is your carboxypeptidase po, which is a group-specific enzyme. So it only cleaves amino acids one at a time. So from the carboxyl end of the peptide chain. Okay. Another naman po is the linkage specificity. Okay? Sa linkage specificity naman po, it acts on the particular type of bond irrespective to the rest of the molecular structure. So very ano po ito, this is the most general type of the specificity of your enzyme. Kasi bonds po exist in all of the molecules in the body, okay? So therefore, so when a enzyme when an enzyme po has a linkage specificity, so lahat po ng may mga type of bond na gusto niya pong i-act upon, yun po, mapaprocess po niya. Okay? For example, your phosphatases po, your can hydrolyze po the phosphate ester bonds in all types of phosphate esters. So hindi niya pipiliin kung anong molecule ito. Basta may phosphate ester bonds, gagalawin niya po yun. Okay? And then for example, glycosidic bonds lang po yung pinaprocess ng enzyme na yon, yung linkage specific enzyme. So lahat po ng may glycosidic bond, is processed by that enzyme. And lastly po is your stereochemical specificity. So such specificity naman po means that an enzyme can distinguish between stereoisomers. Okay? So it can distinguish chiral molecules. Okay? So the chirality is inherent in an active site. So because amino acids po are chiral compounds. So L-amino acid oxidase will catalyze reactions of L-amino acids but not of D-amino acids. Okay. Let's now proceed now to the different factors that affects enzyme activity. So pakitandaan po that these different factors that affects your enzyme activity are also the factors that can affect or denature your proteins. Okay? So enzyme activity po measures the rate of at which an enzyme converts substrate to products in a biochemical reaction. So kung gaano po kabilis i-convert ng isang enzyme ang substrate to its product. So what are the different factors that affects your enzyme activity? So number one is your temperature. Number two is your pH. Number three and four is your substrate concentration and enzyme concentration. So take a look at it uh, at each factors one by one. Okay. First of this would be your temperature. Okay. So how temperature affects your enzyme activity. Okay. So temperature kasi is a measure of kinetic energy of molecules. Now, higher temperatures po mean molecules are moving faster and colliding more frequently. So, ano nangyayari po, the molecules of your enzymes, the proteins inside your enzymes, are moving faster. So, effect po niyan is that the enzyme activity po goes in a boost po in the activity of your enzyme. So, bibilis po yung activity ng enzyme po. Pero take note po that when temperature increases beyond a certain point, the increased energy po begins to cause disruption in the tertiary structure of the enzyme. Thus, denaturation would occur po on the enzyme. So, for human enzymes po, the optimum temperature lang po is around 37 degrees normal body temperature. Okay? So, um, kapag po pumunta na po dun sa highest peak nung enzyme po na temperature, so pagka tinaasan mo palalo yung temperature, hindi na po tataas yung enzyme activity. Instead, bababa po ng bababa yung enzyme activity. Kasi po, nature na, masasira na yung structure ng isang enzyme. Okay? So the optimum temperature is the temperature po at which an enzyme exhibits maximum activity. So pag na-reach na po yung maximum activity, when you succeedingly increase pa yung temperature, bumababa po yung enzyme activity. Okay. Next naman po is your pH. Okay. So how pH naman po affects the enzyme activity? So remember po, di ba, that the amino acids po inside your enzymes have the property called Zwitter ions, meaning they have acidic and basic charges. Okay? So the charge on acidic and basic amino acids are located at the active site. That depends on pH. Okay? Kaya po small changes in pH po can result in the enzyme denaturation and loss of catalytic activity. So that's why po our body po contains biochemical buffers that maintains the optimum pH for an enzyme. Okay. So it can also affect the substrate, causing either um, protonation or deprotonation of groups on the substrate. Okay. So the optimum pH in the body po plays around po 7.0 to 7.5. So neutral po. So in this optimum pH, your enzymes po exhibits maximum activity. So kapag po tinaasan ng pH or binabaan ng pH, the activity po of most of your enzymes po slows down. Okay? So, kaya po lahat na, mostly of your enzymes in the body is at a maximum rate in the pH of 7.0 to 7.5. But some of the enzymes po, like your specialized enzymes like your pepsin and trypsin, can work best on a lower or higher pH. Okay? In the case of your pepsin, it is active in the stomach. That's why it functions best at a lower pH po. Acidic pH na po siya. While your trypsin naman po operates in the small intestine, so it functions best naman po at a higher pH. So, depende po sa environment ng enzyme. But most of your enzymes in the body goes around 7.0 to 7.5. Now, in the substrate concentration naman po, so anong nangyayari po dito, an increase po in the concentration of the substrate will obtain the enzyme activity, not the maximum activity, but the normal activity of an enzyme. Okay? So the enzyme activity increases up to a certain substrate concentration lang po, and thereafter remains constant. Okay? Kasi nga po, your enzyme is not consumed in the process, the substrate is consumed in the process. So kapag po kinonsume po ito, mauubusan po yung enzyme na ipaprocess. That's why magiging constant po yung activity ng enzyme. So the activity pattern is called a saturation curve. Okay? So as some substrate concentration increases, the point is eventually reached where the enzyme capabilities po are used to their maximum extent. Now the rate remains constant from this point on. Now, each substrate po must occupy an enzyme active site for a finite amount of time, and the products po must leave the site before the cycle can be repeated. So, kapag po nangyari yon, each enzyme molecule po is working at a full capacity. Now, the incoming substrate molecules po must wait their turn for an empty active site. Now, at this point, the enzyme po is said to be under saturation conditions. Now, yung rate po at which an enzyme accepts substrate molecules and the release of the products po it is called a turnover number. So, ito po yung time na hinihintay po nung susunod na substrate para po ma-accommodate yung active site ng isang enzyme, the turnover number. Okay? So, an enzyme's turnover number is the number of substrate molecules transformed per minute, okay, by one molecule of enzyme under optimum conditions of temperature, pH, and saturation. So, the next slide po shows you the saturation curve graph of your substrate concentration. Okay. So this graph shows the change in enzyme activity with a change in substrate concentration at constant pH, temperature, and enzyme concentration. Now the enzyme activity remains constant after certain substrate concentration is reached. Okay? So makikita niyo po dito. Okay? So as you increase the substrate concentration at initial, tataas po yung rate of reaction ng isang enzyme. Pero po pagka tinaasan niyo pa po yung substrate concentration, the rate approaches a maximum reaction rate. So hindi po siya tuloy-tuloy na tumataas. Okay. So this slide naman po shows us the relationship of the turnover time to the activity of your enzyme. So pakitandaan nyo lang po that the rate of reaction of an enzyme depends on the complexity of the enzyme reaction or action. Okay? For example, carbonic anhydrase po, it is not complex po in a sense. Okay? Hindi po complex yung pinaprocess niya. It only process kasi a small molecule called your carbon dioxide and water. So, combining it to produce carbonic acid. So, kaya po ang turnover per minute po niya, yung paggawa po niya ng product, is mabilis. No? 36 million per minute po yung nagagawa niyang carbonic acid. So, in the case naman po of your DNA polymerase 1, it is very complex, no? Yung kanyang action. That's why po, maunti lang po yung ginagawa niya per minute. Okay? So, nakadepende sa complexity ng isang enzyme yung turnover time niya. Okay. And lastly, the enzyme concentration. So, paano naman po nakaka-affect ang enzyme concentration in the enzyme reaction rate? So, for it to further understand, you just look at this graph, okay? So, as you increase the enzyme concentration, you also increase constantly the reaction rate, okay? So, nangyayari yan kasi nga the enzyme is kept in a low number because enzymes po are not consumed in the reaction. Kaya po, the greater the enzyme concentration, syempre, the greater the reaction rate din. So, kung patuloy niyo pong tinataasan yung enzyme concentration, tataas lang din ang tataas ang reaction rate po niyan. Okay? Okay. So, this slide shows us lang the existence of what we call your extremozymes. So, these are enzymes that can tolerate, no, extreme environments like beyond the normal temperature pH po and the extreme environments. Okay? So, tinatawag po itong mga organisms na to na merong extremozymes is called extremophiles po. Okay? So, iba-iba ang mga extremophiles. Meron po yung mga acidophiles, alkaliphiles, hyperthermophiles, no, halophiles, and cryophiles. So, they can just exceed the maximum optimum temperatures and pH in the environment. Okay? They can live with that extreme environments. Okay, so let's now proceed now to this topic, okay, called your enzyme inhibition. So the rate of enzyme catalyzed reactions can be decreased po by a group of substances called inhibitors. So the enzymes po kasi are very sensitive to its environment. So the changes lang po in the environment and also existence of inhibitors can hindrance or can hinder its activity and causes it to slow down or to shut down its activity. Okay? So an enzyme inhibitor po is a substance that slows or stops the normal catalytic function of an enzyme by binding to it. So ito, kapag nag-bind po ito sa enzyme, on the different sites on your enzyme po, it can slow down or stop the normal catalytic function of an enzyme. So there are different classifications or types of enzyme inhibition. So meron po tayong reverse competitive inhibition, reverse non-competitive inhibition, and irreversible inhibition. So let's take a look at it one by one. Okay, first of all, this would be a reversible competitive inhibition. So it's governed by a competitive enzyme inhibitor. So when a competitive inhibitor po binds to an enzyme active site, ang nangyayari po dito, the inhibitor remains unchanged, okay? So no reaction occurs, but its physical presence at the site prevents a normal substrate molecule from occupying on the site. So this results po in a decrease in enzyme activity. So paano nangyayari po 'yon? So the competitive enzyme inhibitor po kasi kamukhang-kamukha niya po yung normal substrate po natin. So kapag po yan kamukhang-kamukha ng normal substrate natin, the enzyme active site po is can readily accommodate that inhibitor. Okay? Pero since po hindi po yan napoprocess iyon, iba ang molecular structure nun, parehas lang po yung itsura, okay? It can compete with the substrate for the occupancy at the active site. So ito po, pagka inoccupy niya po yung active site, wala na po mangyayaring enzyme activity. Okay? So it remains po in change as it binds to the enzyme's active site. So ito po, kapagka po nag-bind po on the active site, hindi naman po nang nababago yung shape ng mismong active site. Parang tinatakpan niya lang po um, non-permanently or temporarily yung active site po ng ating enzyme. Okay? So that's why po reversible pa rin ito kasi po weak interactions lang po yung nangyayari between the competitive inhibitor and the active site. So therefore po, pwede po itong i-reverse. Ibig sabihin, pwedeng ibalik sa dati yung enzyme activity po. How? You just increase the substrate concentration, yung normal substrate niya. You increase the concentration of the normal substrate and then you will overwhelm na the competitive inhibitor. So once overwhelmed, matatanggal na po itong competitive inhibitor na to and the enzyme activity po goes to a normal phase. Okay, next naman po is the reversible non-competitive inhibition. So ganun pa rin po ito, it is governed by weak interactions. So therefore po, you can overwhelm the this type of inhibition by a increase in normal substrate concentration. Pero po, paano po naman nangyayari ang isang non-competitive enzyme inhibition? Okay? So, ito naman po, yung ating non-competitive inhibitor, hindi po niya kamukha yung normal substrate. So, hindi po niya kayang maging competitive to the active site in your enzyme. Instead po, it binds to another site in your enzyme. So, tinatawag po yun na regulatory site. Later po, we will learn on that. So, magbabind po yun on the different site in the enzyme. And then, in that binding, it causes naman po a structural change po on the active site. So, binabago naman po niya yung shape ng active site by binding to a different site in the enzyme. Yun naman po, kapag nabago na yung shape ng mismong active site, it will not accommodate the normal substrate. Kasi nga po iba na po yung 3D conformational shape ng mismong active site. So therefore po, lowering or stopping the enzyme activity. Okay? So kaya nga lang po, weak interaction pa rin po yung interaction ng non-competitive inhibitor dun sa regulatory site. Therefore po, you can also overwhelm the the non-competitive inhibitor by increasing the concentration of the substrate. Okay? And then therefore, reversing again the inhibition. So magiging normal ulit po yung enzyme activity. Okay. So lastly naman po is a type of irreversible inhibition. Pag sinabing irreversible na po, the enzyme po can no longer function. Okay? So paano naman po naging irreversible ito? So in your irreversible naman, it includes the irreversible enzyme inhibitor. So this molecule po inactivates the enzyme by forming a strong covalent bonding to an amino acid side chain group at the enzyme's active site. So irreversible na po ito kasi strong bond na po ito. Okay? So, yung irreversible inhibitor, hindi po kamukha ng substrate, pero kapag dumikit po ito sa active site, it does not compete with the substrate. So instead, pag dumikit po yan, magiging permanent na po yung pagdikit niyan. So pag naging permanent, may iba na yung shape po ng mismong active site. So this enzyme inhibitor do not have structure similar to the enzyme's normal substrate. So pag dumikit na yan, magiging permanent na siya. Permanently na rin na mababago yung active site. Therefore, the enzyme can no longer accommodate the normal shape of the substrate. So therefore, irreversible na po or permanent na po um, yung damage dun sa isang enzyme. Okay. So let's now proceed now to the second to the last point of our discussion, which is the regulation naman po of the enzyme activity. So sa katawan naman po, so hindi po pwedeng hayaan ng cells po natin um, na ang isang enzyme po continuously nagwo-work. So, ano po ba ang negative effects ng uncontrolled enzyme activity? So, there are two major effects of an uncontrolled enzyme activity. So, one of it is your wasting of energy, okay? So, some of the enzymes po in the body po uses energy or energy molecule or ATP. So, once that they are not controlled, so patuloy po nilang i-waste or gagamitin yung energy sa katawan. So, mangyayari po, mawawala na po ng energy yung ibang mga processes. Okay? So therefore, the production of the enzyme needs to be turned off. So another naman po na bad result po of uncontrolled enzyme activity is the unnecessary production or overproduction of the products that will later accumulate on the system. So delikado rin po yun. So it needs to be turned off. Okay? So there are many mechanisms po that exist in the body that control your enzyme. Okay? So that turn on and turn off your enzyme. Okay? So this such mechanism is divided into three. So first mechanism po is the feedback control mechanism using the what we call your allostatic enzymes. Second of this mechanism naman po is the usage of proteolytic enzymes, enzymogens. And lastly is the usage of covalent modification. So isa-isahin po natin sila. Okay. So first of this mechanism po is the feedback control mechanism using the presence of what we call your allostatic enzyme. So let's learn first po your allostatic enzyme. So what are these allostatic enzymes po ba? Okay? So the allostatic enzymes po are specialized enzymes po that have two kinds of binding sites. So this would be a binding site for your substrate called your active site and the binding site naman po for regulators or what we call your regulatory site. Okay? So in its structure, allostatic enzymes po have quaternary structures po there. They have two or more protein chains. Okay? Now the active and regulatory binding sites po are very distinct from each other in both location and shape. So the binding of a molecule at the regulatory site causes po changes in the overall 3D structure of the enzyme. So it includes structural changes at the active site also. Okay? So in this binding of the regulatory substance or regulator po in this type of regulatory binding site, it can change its shape, the shape of the enzyme and also shape of the active site, therefore po controlling its activity. Okay? So there are two types of allosteric control. Pag sinabing allosteric control, you use allosteric enzymes to control the enzyme. Okay? So what are these? So first of the allosteric control is your positive allosteric control. So let's take a look at an allosteric enzyme. So dalawa po yung site niya. Meron po siyang active site and regulatory site. Okay? So sa isang positive allosteric control, you use positive allosteric regulator. So when in the presence of this positive allosteric regulator and binding of this substance po to the regulatory site, it will cause a conformational change on the active site, allowing the entry of the substrate po on the active site. When it allows the entry of the substrate po, the enzyme po is turned on. Okay? So, active na po yung mismong enzyme to function. Okay. Another allosteric control naman po is a negative allosteric control naman. Ito naman po yung ginagamit ng cell to turn off the activity of your enzyme. So, using a negative allosteric regulator. Okay? So, tinatawag din po na, please take note na lang po na tinatawag din po yung regulatory site na allosteric site. Okay? So, upon the binding of a negative allosteric regulator on the allosteric site, ano naman po ang nangyayari? Now, the normal shape of the active site is then changed to a abnormal active site. So it cannot now accommodate the substrate normally. So therefore, the enzyme po is turned off in the process. Okay. So again po, your allosteric control is governed by your regulators, no? Ito po ulit yung mga substance that binds on the regulatory site or the allosteric site of an enzyme. So meron po again tayong positive regulator and negative regulator. So it means that a positive regulator increases enzyme activity. So ang nangyayari, the shape of the active site is changed so that it can more readily accept the substrate. And then the negative regulator naman po decreases the enzyme activity. So it changes the shape of the active site to make the substrate po less readily accepted by the active site. Okay. So one of the mechanisms by which your allosteric enzyme activity is regulated po is your feedback control. Okay. So feedback control is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of reaction sequence. So there are different types of feedback control. Meron po tayong positive feedback control and negative feedback control. So dito po sa feedback control, the concentration of the final product or the final product of the reaction po um, can control po the whole um, production of the product. Okay? So for example, on the negative feedback control, so at a low concentration of the final product, the D product, the reaction sequence proceed rapidly. Okay? So kapag low concentration ito, normal po yung activity. Pero kapag tumaas or in higher concentrations of the D, letter D product, now the activity of enzyme 1 becomes inhibited. Okay? So nangyayari nito, the final product can inhibit the first enzyme, okay, by overwhelming the enzyme po or inhibiting or binding on the regulatory site po, and then therefore turning off the enzyme 1. Okay, so eventually the activity loss. Okay, so please take note that the feedback control is not the only mechanism by which an allosteric enzyme can be regulated. So it's just one of more common ways. Okay, so regulators po of a particular allosteric enzyme may be products po of entirely different pathways of reaction within the cell. So do you know also that compounds outside the cell or produced outside the cell can be regulators of the enzymes. Examples of these are your hormones. Okay? So hormones po, 'di ba, govern the some of the most of your system po. Okay? So when hormones are present in the bloodstream, um it is accepted by the cell, and then pag pumasok po sila sa cell, it can inhibit or it can bind to the allosteric site or regulatory site, turning off then the enzyme activity. Now, a second mechanism for regulating the cellular enzyme activity is based on the production of enzymes in an inactive form. Okay? So these inactive enzyme precursors are then turned on at the appropriate time. So such mechanism po for control is often encountered in the production of proteolytic enzymes. Okay? So your proteolytic enzymes po catalyzes the breaking of peptide bonds that maintains the primary structure of a protein. So they are generated in an inactive form and converted to active form when they are needed. So anong tawag po dun sa inactive form or inactive precursor ng isang proteolytic enzyme? Now, the inactive precursor of a proteolytic enzyme is called your zymogen. So activation of a zymogen requires an enzyme also. Okay? So it's controlled reaction that moves some part of the zymogen structure, which changes the 3D structure of zymogen. So which affects its active site conformation. So upon the presence um of another enzyme that activates their zymogen, so your zymogen can turn into an active proteolytic enzyme to stop the process. Okay? So let's see on the other slide kung ano po yung itsura ng isang zymogen at isang proteolytic enzyme. Okay. So this slide po shows us the conversion of a zymogen, an inactive form of a proteolytic enzyme, to a proteolytic enzyme, which is the active form of the enzyme. So this often involves removal of a peptide chain, ito po, segment from the zymogen structure. So once po na na-remove po ito, naging active na po yung isang enzyme. Okay? So nagkakaroon na siya ng isang active site to work on to other enzymes also, to control other enzymes. Okay. So last mechanism po to to control your enzyme is the covalent modification of the enzyme na, okay? Kapag sinabing covalent modification, so this is a process in which the enzyme activity po is altered na by covalently modifying the structure of the enzyme. So how do you covalently modify the structure of an enzyme? So this is through the attachment of a chemical group or removal of a chemical group from a particular amino acid within the enzyme structure. So the source of the added phosphate group, which is often an ATP molecule, okay? So means um most of the time po, phosphate addition of the phosphate group po can be a source of modification of the structure of an enzyme, okay? So the process of addition of the phosphate group to the enzyme is called phosphorylation. And the removal naman po of the phosphate group from the enzyme is called dephosphorylation. And these two processes po, if it happens po inside the cell, it can control your enzyme activity. Okay. So phosphorylation again is the addition of a phosphate group to the enzyme by protein kinases. Okay. So, di ba po kanina we have learned the different classifications of your enzyme. So, one of which is your kinases. Now, the phosphorylation naman po is the removal of a phosphate group from the enzyme by your phosphatases naman. So, and the last point naman po of our discussion, so we will discuss naman po the importance of your enzyme in the diagnostic procedure or in the laboratory. So, your enzymes po can be medically used po to diagnose certain diseases, okay? So, although blood serum po contains many enzymes, so some enzymes pa rin po are not normally found in the blood, but are produced only inside cells of certain organs and tissues. Now, the appearance of these enzymes po in the blood often indicates po that there is tissue damage in an organ and that cellular contents are spilling out into the bloodstream. So, this is used in the treatment of diseases. Okay? So, enzymes can also be used in the treatment of diseases. Okay? So, recent advancement in treating heart attacks is the use of tissue plasminogen activator, which activates the enzyme plasminogen. So, when activated, this enzyme dissolves blood clots in the heart and often po provides immediate relief. So another medical use of four enzymes is in clinical laboratory chemical analysis. For example, so no simple direct test for the measurement of urea in the blood is available. However po, if the urea po in the blood is converted to ammonia via the enzyme urease, the ammonia po produced, which is easily measured, becomes an indicator of urea. Now, this blood urea nitrogen test is a common clinical indicator or laboratory procedure. So, high urea levels in the blood indicates kidney malfunction. So, yan po yung mga different uses of your enzymes. So, yan po, kung makikita po ninyo in this table, so show us different presence of enzymes into different tissue types. Okay, for example, in heart and liver. So, your lactate dehydrogenase can exist. Okay, kaya po in the appearance of this type of enzymes in the serum can depict as different conditions po in the patient. Okay, so yan. So for example, your CPK or your creatinine phosphokinase can indicate alone heart disease because siya lang po yung nag-exist na enzyme inside the cells of your heart.